In this video, we're going to be talking about the clinical correlates of the pyrimidine synthesis pathway. So in the previous video, we talked about pyrimidine synthesis pathway in more detail, and we also talked about the role of folate and B12 in pyrimidine synthesis. In this video, we're going to be talking about the clinical correlates associated with it, and we're going to talk about the pathologies that are associated with abnormalities in the pyrimidine synthesis pathway, and also drugs that we use that target the pyrimidine pathway. Okay, so there's only one pathology that is associated with pyrimidine synthesis pathway, and that is going to be orotic aciduria. Okay, so this is an autosomal recessive defect in which there is a deficiency in the enzyme UMP synthase. Now, if you recall, when we were talking about the pyrimidine synthesis pathway, orotic acid is combined with PRPP to form UMP, and this is going to be catalyzed by the enzyme UMP synthase. Now, if you have a defect in UMP synthase, that's going to lead to a buildup of orotic acid in the blood, and of course, orotic acid is then going to be excreted in the kidneys into the urine, hence the name orotic aciduria. There's also going to be a abnormality in pyrimidine nucleotide synthesis, and that's going to lead to other clinical findings like megaloblastic anemia and also growth retardation. The treatment for this pathology is going to be to give the patient uridine. Uridine is going to be converted in the body directly to UMP, bypassing the need for UMP synthase enzyme. So now we're going to talk about the drugs that target the pyrimidine synthesis pathway. These drugs are going to be Ara-C, hydroxyurea, 5-FU, methotrexate, antibiotics, pyrimethamine, and leflunomide. The drug that we're going to talk about is called Ara-C. Now, this is a chemotherapy agent. This drug mimics the structure of cytidine. So this is going to be the structure of Ara-C, and this is going to be the structure of cytidine. As we can see, they have very similar chemical structures. So this drug's mechanism of action works as a pyrimidine analog. So when this drug enters the cell, it's going to bind to DNA polymerase, and it's going to inhibit it. And of course, it's going to prevent DNA synthesis inside the cell, and that's going to lead to cell death. So the next drug that we're going to talk about is hydroxyurea. Now, hydroxyurea's mechanism of action is that it inhibits the enzyme ribonucleotide reductase. Now, remember that we said in the thymidine synthesis pathway that UMP is going to be converted into UDP, and UDP is going to be converted into deoxy-UDP by the enzyme ribonucleotide reductase. So hydroxyurea is going to inhibit this enzyme, and thus it's going to inhibit the synthesis of thymidine. Now, this drug can be used for certain malignancies like polycythemia vera and essential thrombocytosis, but it is rarely used for malignancy. It has a special use is that it can be used in sickle cell patients because it's going to increase the fetal hemoglobin levels. So the next drug that we're going to talk about is 5-FU, and its main mechanism of action is that it is a uracil analog. So when this drug enters the cell, 5-FU is going to be converted into 5-FDUMP. So it has very similar structure to deoxy-UMP. And of course, that gives it the ability to bind to the enzyme thymidylate synthase and inhibit it. When this enzyme is inhibited, that's going to prevent the formation of deoxy-TMP or thymidine. And of course, that's going to lead to cell death. The next drug is methotrexate. Methotrexate could be used as a chemotherapy drug, and it could also be used as an immunosuppressant. So the mechanism of action of methotrexate is that it mimics the structure of dihydrofolate, and that gives it the ability to bind to the enzyme dihydrofolate reductase and inhibit it. When dihydrofolate reductase is inhibited, that will decrease the amount of N5-N10 tetrahydrofolate that is available, and that is thus going to prevent the synthesis of thymidine. The next drug that we're going to talk about is called leucovorin. Now, this drug doesn't have anything to do with the pyrimidine synthesis pathway, but it's important to know because it's always given in association with methotrexate to prevent bone marrow suppression. Now, we said before that methotrexate is going to inhibit the enzyme dihydrofolate reductase, and that's going to, of course, decrease the levels of N5-N10 tetrahydrofolate. So what leucovorin does is that it's going to be converted inside the body into tetrahydrofolate, bypassing the need for this enzyme. And of course, that's going to prevent bone marrow suppression in these patients. So we have certain antibiotics that target the pyrimidine synthesis pathway in bacteria. These antibiotics are going to be the sulfonamides and the trimethoprim. So it's important to know that humans get their folic acid from the diet. Now, bacteria can't absorb folic acid, so they have to synthesize it. And they synthesize it from the molecule PABA. So bacterial cells make PABA, and they convert it to dihydrotoroic acid by the enzyme dihydrotoroate synthase. Dihydrotoroic acid is converted to dihydrofolic acid, and dihydrofolic acid is converted to tetrahydrofolate by dihydrofolate reductase enzyme, and that of course is going to help in the synthesis of thymidine. So sulfonamide antibiotics inhibit the enzyme dihydrotoroate synthase, and trimethoprim inhibits the enzyme dihydrofolate reductase, and of course this is going to inhibit the ability of bacteria to form DNA, and that's going to lead to bacterial cell death. Okay, so pyrimethamine is used to treat parasitic diseases. So the mechanism of action of this drug is that it inhibits dihydrofolate reductase in certain protozoa, and of course that inhibits the DNA and RNA synthesis in these protozoa, and that leads to their death. Okay, the last drug that we're going to talk about is called leflunomide. Now, leflunomide is an immunosuppressive drug that is used in rheumatoid arthritis. So how does it work? So remember, in the second step of uridine synthesis pathway, we convert carbamoyl phosphate to orotic acid. Now this step is going to be catalyzed by the enzyme dihydroorotate dehydrogenase, and of course an aspartate is going to be used in the process. So the leflunomide inhibits this enzyme and prevents this reaction from going forward, and of course that's going to inhibit pyrimidine synthesis. Leflunomide mainly affects active T cells, so it's going to prevent the proliferation of active T cells, and that's what leads to its immunosuppressive effect.